Bene, buongiorno a tutti, vi invito ad accomodarvi e benvenuti a questa seconda giornata di Trieste Next, una giornata soleggiata ma parecchio ventosa, per cui per tutte le persone non di Trieste che avessero avuto piacere di sperimentare cos'è la bora, oggi avete una netta dimostrazione, anche se non è proprio la bora fortissima che c'è di solito in questa città. Vado brevissimo, così diamo subito spazio ai nostri ospiti di questa mattina. Quando abbiamo iniziato a costruire il progetto di Trieste Next qualche mese fa, abbiamo trovato subito un interlocutore privilegiato. Sin dalla prima edizione, infatti, Trieste Next si vuole proporre come una manifestazione internazionale. Quindi siamo particolarmente felici che prima la Commissione Europea, rappresentanza di Milano, e poi il Consiglio Europeo delle Ricerche e lo European Research Council abbiano deciso di promuovere e patrocinare questa manifestazione. Quindi un ringraziamento a Massimo Gaudina, il direttore della rappresentanza di Milano della Commissione Europea, un ringraziamento ad Angela Liberatore, la responsabile appunto del capo unità Social Sciences dello ERC, del Consiglio Europeo delle Ricerche, e grazie anche a Rosaria Carbone, responsabile della comunicazione dello ERC. È una testimonianza fondamentale quella di questa mattina, mi permetto di dire, perché è un'attestazione del valore internazionale di questa manifestazione, ma soprattutto perché vogliamo proporre 4 ricercatori che sono una rappresentazione dell'eccellenza europea della ricerca, eccellenza che è fatta di interdisciplinarietà, di diversità non solo di genere. Fortunatamente abbiamo 3 donne con noi sul palco oggi e un uomo, ma anche diversità di culture, di linguaggi, di discipline scientifiche. Questo è un tema che sin dalla prima edizione di Trieste Next noi vogliamo portare avanti e appunto avvalorare, e do il benvenuto ad Angela Liberatore. Grazie ancora una volta, buon ascolto e lascio a te la parola. Grazie mille.
Grazie Antonio. Ovviamente la bora è un piccolo caso di necessità di adattarsi a cose non molto previste. Prima domanda, alcuni relatori parleranno in inglese e altri possono parlare o in italiano o in inglese. Levata di mano, possiamo fare tutti in inglese? Opzione 1, o qualcuno preferisce un po' e un po'? Per tutto in inglese? Bene, grande maggioranza, perfetto. Quindi sarà, so we shift immediately, it will be everything in English. So I would like to ask our guests, Agnieszka Wikowska, to be with us. Applause for Agnieszka, please. Agnieszka is principal investigator. She comes to us from Poland after being in several other countries, including Sweden and Germany, and she's now at the Italian Institute of Technology. She will tell us more about her ERC grant. Please take a seat. We have Laura Concetta. Applause for Laura. Laura is also at the Italian Institute of Technology. She has been not always in Italy, actually many places, including Berkeley and others. And she will tell us more about her research, and I will introduce her again when it's the time to speak. Thank you, Laura. We have Roman Nierala. I'm not sure the pronunciation is perfectly good. Roman comes to us from Slovenia. Actually, you were driving this morning from Ljubljana, our great neighbors, and he has been traveling as well in his scientific endeavors, but he's based in his home country, Slovenia, for the ERC grant. And last but not least, we have Silvia Marquesan, who I guess you all know. Silvia is another case of excellent research, in this case not funded by ERC, even if she says she aspires to in the future. But the session is about celebrating excellence anyhow, so you're most welcome. And actually, the project that Silvia is coordinating is based on an Italian program which is modeled on the ERC one. So I think that we are definitely, not putting boundaries on who funds what when the matter is excellent. So let's get started. Our speakers have around 12 minutes to present their work. After that, I will, ask an ice-breaking question for them to think about some more transversal issue, and you can think about any question you might have to any of them. You will be able to raise your hand, get a microphone, and ask the question. And at 11:15, we need to close. Okay, this is for the proceedings. Now, the first, then in alphabetical first name order is Agnieszka. Agnieszka is a principal investigator that works on social cognition in human-robot interactions. She has actually a background in philosophy and psychology, and then started working in a very interdisciplinary way on this issue of human machines, robots interaction, which is very close to the topic of Trieste Next this year. As I said, she was in other countries, and then she's actually one of the very few cases where Italy attracts foreign scientists to Italy, because unfortunately, the majority for the time being is that Italian scientists are working abroad, but surely this can be improved in the future. So Agnieszka, the floor is yours.
Thank you very much. Thank you for inviting me here. I would like to tell you a bit about my research and where we're aiming with our studies at Italian Institute of Technology. So I'm interested in humans interacting with robots. And when you think robots, many of you might have something like this in mind.
R2-D2, where are you? At last, where have you been?
So these are robots that are very good in walking around and being worried and having emotions, apparently. He's worried, where have you been? And it's quite an advanced robotic system. Or you might also think of something like that. Extremely human-like robots.
I'm Caleb.
Hello, Caleb.
So as you see, this robot here is depicted as being very, very human-like and also being able to process speech, respond appropriately to the point that it's almost indistinguishable from a human. Well, that's our fantasies, but in reality, the present robots look more like this and behave more in this way. So we're apparently not there yet. And the reason why we're not there yet is because it's an extremely complex problem to build robots that can navigate in our environments the way we do, that can interact with us naturally, and that can just sort it all out in the way humans do. So that's kind of state of the art, but we would like to be in this situation where we would have robots helping us out in daily lives, in daily course, as you can see in the upper pictures, being our assistants, perhaps in supermarkets, being also of assistance in healthcare and childcare. And that's our dream, and we strongly believe that robots can be extremely helpful in the future, but as I said, we're definitely not there yet. So what do we need to do to get there from where we are? Of course, there's a massive effort that needs to be done on the engineering side and on the AI side. We need to work on all aspects of AI and robotics, both software and hardware, to make that happen. But me, myself, I'm not a roboticist, so I will tell you about something else that also needs to be done, and that is the human side of human-robot interaction. Since my background is psychology and cognitive neuroscience, I'm interested in actually what sort of cognitive mechanisms are evoked by a robot when a human interacts with a robot, because we need to design those robots in such a way that humans respond to them as if they were social entities, and that it's intuitive for a human to actually interact with a machine like that. And this is where our research comes into play. I often use the term social attunement. Social attunement is a concept that is kind of an umbrella concept that subsumes all possible mechanisms of social cognition that we use on a daily basis with one another. So there are very often implicit, very automatic mechanisms, and in the next video you will see I'll demonstrate a subset of such mechanisms that we use. Okay, so in this video demonstrates such, as I mentioned, mechanisms. So pointing gesture is something that we use all the time to communicate our intentions. And then the question is how to make these things intuitive so that humans understand the communication that is displayed by a robot, or that the robot understands the pointing gesture or other types of gestures that the human communicates. Then there's a level of joint action, interaction. There was mutual gaze at some point, which is a very important aspect of human communication, and I'll talk about this later in a second. There is something that is called emotional synchronization, you might say, because when we're successful in our tasks together, we're happy together, and the synchronization part is very, very important as well. So this is just a demonstration of the mechanisms that we're investigating, and I'll tell you a few examples in a second. So how do we need to approach this problem? As I mentioned, my background is cognitive neuroscience, and we use cognitive neuroscience methods and methods of experimental psychology to actually understand how the brain processes information during interaction with a robot. So this is just an example setup of our experiment. What you see here is a participant of our experiment interacting with the robot, handing over objects, and while she's doing that, we're measuring her reactions by a mobile eye tracker. For example, these are just example methods that we're using. So a mobile eye tracker that tracks the movements of the eyes while a participant is doing a task. And that tells us a lot about, for example, attentional focus, whether she's attending the right elements of the scenario and whether she would be engaging similar kind of behavioral mechanisms as in the case of interacting with a human. Then sometimes we also, or most of the times actually, we give our participants a task in addition to the interaction, and we measure their performance, which is measured very often with reaction times or error rates in a task that tells us about how well those cognitive mechanisms have been employed in an interaction. And finally, this is an EEG cup, electroencephalogram, which measures brain activity during a task. And that can tell us a little bit about the neural mechanisms that are underlying the behavioral mechanisms. So through these kind of designs, we try to understand the brain and behavioral responses of the human in an interaction. Now, the research questions, this is just a simple subset of questions that we're asking, but I chose those because I thought they were maybe most interesting. The research questions that we're asking are, for example, do humans engage in joint attention with a robot? What is joint attention? Joint attention is a situation that if I point there and look there, probably you will attend there as well. It's a very automatic process, and it underlies most of social cognitive mechanisms that we have. It's very important. Little babies learn that very fast, and it's impossible basically to engage in a social interaction without having that mechanism. So it's very important for us to understand how to design the robot behavior in such a way that a robot would engage our participants in joint attention. Because if we can't achieve this, then the interaction will not be smooth and successful. So this is a question that we're asking, and our findings show that indeed, under certain well-designed behavioral circumstances or behavioral characteristics of the robot, humans would engage in joint attention. And one of them is eye contact, something that I already mentioned, very important. So if the robot engages our participants in eye contact, then they're willing much more to engage in joint attention later on. And this is just an example of our robot looking into the eyes of participants. This is from the participant perspective. And this is thanks to an algorithm that this robot, that has been, by the way, designed, this is ICAB robot, designed at the Italian Institute of Technology. That robot has an algorithm to detect where the face of participant is, to detect where the eyes are, and then direct its gaze towards the eyes. And it's a very, very strong effect. We talk to our participants. They really feel very engaged once the robot does that. So this is already one piece of information that we can then give to our roboticists and tell them, oh, if you want to have a smooth interaction, please make sure that the robot engages your participants in eye contact, as one of many other mechanisms. Another question that we are and this is, this is just to add that we also have measured the neural activity during joint attention and eye contact, and we have observed that actually when people engage with the robot in joint attention, the brain mechanisms are exactly similar to those that they would engage in human-human interaction. So that's very good. That shows that people can treat a robot in a kind of similar social way as another human would be. Another question that we're asking is, can people, and under what circumstances, attribute mental states to a robot? Mental states such as intentions, beliefs, desires. Would you explain a robot behavior in terms of, oh, the robot looked at me because it wanted to communicate something to me? The wanted part is a mental state. We typically use these sort of explanations of behavior towards other humans. But would we do that towards machine? I mean, you don't necessarily explain behavior of your car, oh, it braked because it wanted to brake. You know that it braked because you pushed the brake pedal, right? So humanoid robots are somewhere there in between. If you think of a coffee machine, it's not that it didn't pour coffee because it was angry at you, it didn't pour coffee because it was broken, right? But humanoid robots are in between, and the question in between because they look human-like and they behave more human-like. So would we attribute kind of mental states to robots like this? And our data show that dependent on the circumstances and the context, people actually are capable of attributing mental states to robots. So what we have done is we have developed a set of scenarios like these with 3 pictures that kind of tell a story. And then we ask participants to choose between two descriptions of what's happening there. And you see, one is more mentalistic, so we use kind of mental states. ICAP was trying to cheat, that's a mental state. Or a very mechanistic one, ICAP was unbalanced for a moment. And in this case, if you look at the scenario, it's really sort of provokes you to choose the mentalistic description. This is what actually our participants do. In some scenarios, they are very much willing to attribute mental states to the robot. And we also have measured EEG, and in the EEG signal we see that there are differences actually between different types of attributions, mentalistic or mechanistic. One final example that I wanted to give here is, would we be able to lie to a robot? Because if so, that means that we treat it as a social entity. While, I mean, you wouldn't have a problem with lying to a coffee machine again because it's just a machine. But would we be able to lie to a robot? And in our experiment, we had participants interact in a kind of Skype interaction with ICAB, and they were asked to lie every now and then to the robot. And we measured their heart rate and galvanic skin response. And we saw that, again, it was the eye contact that mattered, because after ICAP looked at participants had much harder time in lying to it. So again, it shows that people are able to treat it as kind of a social entity. So this will hopefully help us design robots of the future that will be well attuned to us and will have impact on not only on robotics, but also on robots for education and for clinical applications, such as robotic therapy for children diagnosed with autism or assistance in elderly care. Finally, I would like to thank the ERC for being able to conduct this fascinating to me research, and my team at IIT, and thank you very much.
Thank you, Agnieszka, while we were here last night about revolution, I think Agnieszka is giving us some hints on many of the revolutions that robotics can bring, including on us as human, an anthropological change. Okay, we are then moving from cognitive sciences to a little bit of a different field, somebody who has been working in the field of medical chemistry and then biology, so again, interaction between different fields of research. Laura Cancedda, who got her graduation in Genoa and then the PhD at the Scuola Normale Superiore in Pisa, and has been traveling in various places, including in University of California, as I mentioned before. So Laura, the floor is yours to present your research, which has also a link with Agnieszka about application to health.
Thank you. So thank you for the invite, and today I'm also going to talk to you about what we do in the lab, which is basically trying to find new therapeutic approaches to rescue cognitive impairment in neurodevelopmental disorders. And we do that trying to, stick to reality and to have an impact on the life of patients, but also pushing our boundaries and try to do very new research. So we're sort of juggling among the present, the future, and since I got my ERC grant, the futuristic. So neurodevelopmental disorders are caused by very, very different etiologies. They give origin to very different disease, but the funny thing is that many of them, they share a number of symptoms, like defective brain development, cognitive impairment, sleep disorders, and increased susceptibility to seizures. And unluckily, they also share the fact that this is the current treatment options for many neurodevelopmental disorders, like nothing. And the only thing that parents are left with when their kids are diagnosed with neurodevelopmental disorders is early educational intervention that can do some good things later in life, and they do highlight the fact that if you intervene early in development, you can have positive outcomes. So there are no pharmacological treatment, but there are animal models, and researchers look into these animal models to try to understand how the disease origins. So this is just to have everybody on board, just a few things about how the physiological brain works. I would say that you can divide the neurons basically in two big families, what are called excitatory neurons, so the one that allows the flux of information, and they release glutamate to talk to one another, and then inhibitory neurons that are called GABAergic neurons because they release GABA, and they sort of give a pace to the information in the brain. So if you want to make it simple, the glutamate is the green signal, information flow, and the GABA is the red signal, like stop. And it's known that to work properly, the brain needs a perfect balance between excitation and inhibition. And we study Down syndrome, okay, which is a neurodevelopmental disorder caused by the triplication of chromosome 21st, so they have an extra chromosome 21st. And on animal models, when we entered the field, what was known was that there is one gene in chromosome 21st that is responsible for production of extra number of inhibitory neurons. So allegedly in Down syndrome, you have too much inhibition. This is why maybe the animals and people with Down syndrome don't learn too much, because it's like they have too much inhibition. They're slower, and they have difficulty in learning. But then I came from the GABA field, and there was something that was a little weird. So people with Down syndrome and animal models, they have increased susceptibility to seizure. They're hyperkinetic. They have anxiety, and all these symptoms, they mostly relate to excess of excitation, not too much inhibition. So that was a little bit of paradox. And we thought that maybe the too many interneurons, the too many inhibitory neurons, are playing an opposite role. So it's not that they have too much inhibition, but GABA is working in the opposite way. So maybe the brain is not working well because you have too much excitation. And there is a way to prove it, and you can do brain slices of your animal model of Down syndrome, like in this example, and do what they're called electrophysiological recordings. It's just a way to listen to neurons that talk to one another. And when excitatory neurons talk, you see these spikes here. So this is like one word of two neurons that are talking. And you see that in animals with Down syndrome, they talk more. And what was interesting for us was that if you give GABA to this brain slice, of course, in neurons that do not have Down syndrome, they stop talking because GABA inhibits the flow of information. But in neurons with Down syndrome, they talk way more. Look at how many words you have here. And we knew, and we were lucky enough, that there was a drug, a diuretic drug, FDA approved, which is called bumetanide, that is being used in the clinics as a diuretic that can change GABA. When it's wrongly excitatory, it can make it back inhibitory. So if you give GABA plus bumetanide, GABA returns inhibitory in Down syndrome, and neurons stop talking. So was this enough to rescue cognitive impairment in animal models? So you see, there is a way to measure cognition in mice. And in this test here, if the video works, you see that you can take an animal, take it from the cage, put it into an arena where it will find two objects. And a curious animal will explorate both objects. Then if you repeat the experiment on the next day, and on the next day you move one object, an animal that remembers will spend most of the time exploring the moved object. An animal that do not remember will explore the two objects at the same way. So you see here, this is learning index. The animal with Down syndrome perform very poorly in this task, but if you give them bumetanide, so you restore GABAergic inhibition, they perform perfectly. So this was the present. This is the next future. So we're going to start a clinical trial, hopefully soon, in collaboration with Bambino Gesù to try whether bumetanide works in people with Down syndrome. But then, at this point of the story, I needed to think about my ERC grant. And I thought that a cool project that would stem from this research would be, for example, to try whether bumetanide can also rescue other symptoms of Down syndrome, like anxiety and sleep, that they do depend on GABAergic transmission. And I really like this project here. Another aim of my possible ERC grant would be to fund a drug that is not diuretic, but it still works on the brain, like a new drug that is better than bumetanide. I thought that this project was very cool, but ERC is about cool stuff, but it's about very innovative stuff. Before it was called ideas, it's really about pushing the boundary of your research. So I did like this project, and I submitted to the telethon, and we're working on it. But for ERC, I thought that I should go a little wilder. And I thought that I should address, at least in my mind, the ideal treatment for neurodevelopmental disorders. And what could that be? Something that starts as early as in the embryos, because the idea is that then you can treat the embryos and you can sort of correct the developmental trajectory into something that maybe as an adult will not need a pharmacological treatment, ideally. So it should be, although something that spares the mother and directly target the fetuses. And this cannot be a normal drug like bumetanide because it's taken orally. And if the mother takes the drug, it will take the drug, right? So the other point it has in the ideal world, it should be brain specific because you want only to target the brain. And for example, bumetanide is very safe in adult subjects, but it was given in a clinical trial to infants, and it caused deafness because it interfered also with the development of the auditory system. So you can't do that with standard drugs. And then it has to be something that selectively repaired the gene that you want to target so that the effect will be very clean and not side effect. And here you go. This is a gene edits, which stands for gene editing in Down syndrome. That is my ERC project. And I like to think of it as, if it works, it will be the proof of concept that the ideal treatment may be at least conceivable in the future. So it has three objectives. In objective one, we will use the state-of-the-art gene editing technology by which you can really cut and repair the genes that are not working well in your neurodevelopmental disorders. And we're going to try to do that in Down syndrome by deleting just one copy, because they have 3 copies, of the genes that are important for cognition. We are going to test the outcome at behavioral level in animal models, and in a way that entails using a technique that we developed, we helped to develop in the lab in recent years, that allows you to target only the fetus and not the mother, and then by strong electric field allows you to target only the brain, not other organs, and only the part of the brain that is important for memory. And then there is another objective to try this technique, not on only one mouse model of Down syndrome, but many others, to see whether it really works on Down syndrome, and in cells derived from the fibroblasts, from the skin of people with Down syndrome, to try to see whether also in human cells would work. And then there is the last aim that is about developing a technology that will allow us not to use very strong electric fields that you can imagine you don't want to use even in a very far future in humans, but ultrasound, which is something that is clinically used every day by pregnant women that go and do imaging of their fetuses like this. But this is, of course, like even beyond the futuristic. And I would like to really thank the people that helped me to push towards the future in the lab, which is my fantastic team in the lab, and the funding agency, and in particular the ERC. Thank you.
Thank you also to Laura, to excellent presentation, and I think both engaging. I'm not a cognitive scientist and even less a chemist or a biologist, but I found them very interesting. Now, the challenge will be to be able also to follow Roman Njerala at the National Institute of Chemistry in Slovenia. Roman is a biochemist and synthetic biologist, so again, somebody with quite a variety of backgrounds. He will tell us about proteins, and again, we will be looking forward to hear from you. And his ERC grant is in the making, so we will learn more about it. Thank you, Roman.
Thank you very much for the invitation. It's a great pleasure to talk in front of the full hall, particularly with so many young people. So I hope you'll be interested in the project that we started to work at our lab. So let me just put it here. So the cause of my main inspiration is really the nature. So nature provides fantastic devices that spans over 10 orders of magnitude from exciting molecular machines that are far beyond the reach of any our nanoscale objects. They are highly efficient, also renewable, but also nature is able to span this to much larger scale. And the molecules that really do this are to a large extent proteins. So proteins are one of the examples of probably the best smart nanoscale machines, because all the function, the tertiary structure of the protein is encoded in just linear sequence of amino acids. However, the rules that govern the folding of this sequence into this 3D object are very complex. They evolved over billions of years of evolution. They are governed by large number of weak, long-range interactions. They define those molecular machines. However, at our macro scale world, we are able to make buildings in sort of two different approaches. One is sort of artisanal, unique, which is very beautiful, very nice. But on the other hand, we built our houses in a modular, engineered way. They're not so nice as those objects. However, this modularity, this modular principle of constructions allows us to make objects that just could not be made by something, by carving. So our idea was to try to apply that type of principles at the nanoscale, at the scale of building molecules. You may know that DNA nanotechnology has repurposed DNA for something completely different. In the last about 30 years, researchers are able to fold DNA into almost any selected shape. As you can perhaps guess what type of country is this made of DNA. So we thought that, okay, DNA is fine, but really proteins are those machines, those molecules that are running the cells. So we wanted to translate something from DNA, from sort of modularity. So if you compare here the compact, normal folded proteins and DNA to the modular fold, so we thought that it would be great to do something like that also on proteins, namely to make well-designed modular structures that are well defined and engineered. By this way, we would be able to bypass the problems of controlling all those weak long-range interactions in proteins. So in order to do, we need to have some building blocks, similar as DNA. As you know, with DNA, it's the complementarity between base pairs that defines which strand binds to another one always in the antiparallel direction. So we, in fact, found that there's something similar in proteins. In proteins, there are coiled dimers, which are the protein helices. They are seen from sort of schematic way from the side and here from the top down. So there are two helices that wind around each other. And the rule of their recognition is quite simple. They're based on hydrophobic interactions here at positions A and D, and electrostatic interactions at those positions. So, in fact, that type of repeat defines which type of peptide should bind to another one. As an additional bonus, we are able to define them in parallel or anti-parallel direction, which, as you will see, is quite important for our strategy. So, nature uses those quite often, so about 5% of proteins contain called building blocks, and they are characterized by this heptate repeat, which spans about 1 nanometers. And in the nature, there are building blocks that span from 3 up to 50 and more nanometers. So we could have several of those building blocks to start from. And the whole idea was the following. The idea was to select those coil complementarity building blocks and concatenate them together in a single long polypeptide chain, here connected by flexible linkers that act like hinges. So in this case, in this chain, the blue segments are complementary, so they will come together and form a rigid coil dimer, and this will seed the formation of the final three-dimensional structures. And what we want to do with those structures, the idea is to design different types of polyhedra. As you know, with polyhedra, you can basically, in principle, make almost any shape you want. So in this case, we just draw the outline of the shape, and then we have to design the part that goes and makes a stable structure. So here is one example of a chain that we just drew on a paper, so it's not much of modeling that's required for this purpose. As you can see here, this tetrahedron starts and ends in the same vertex, and it's composed of antiparallel, you can see arrows in different directions, and 4 parallel segments. It's a single long polypeptide chain. In fact, we realized that that's in fact a sort of a mathematical question. Perhaps you remember Leonhard Euler, about 300 years ago, founded the theory of graph theory when he wanted to solve the famous riddle of bridges of Königsberg. The question was, can you traverse each of those bridges by in a single path going only once over one bridge? And of course, Euler proved that that's impossible. But on the other hand, if we look at our tetrahedron, you can see that we have to connect each vertex across exactly twice. So Leonhard Euler tells us that it is possible to make such a polyhedron. In fact, the mathematicians that were quite excited to help in this project proved that it's possible to make any type of selected polyhedron, regardless of complexity, just by a single polypeptide chain. So that was the theory, but the experimental implementation was simply by selecting the building blocks. Some of them we designed, some of them were taken from the nature, and those are the codes for those segments. So APH segment should form an anti-parallel dimer with the fifth segment. The second segment should form parallel dimer with the eighth segment. Here you can see P3 with P4 and so on. So that's a single polypeptide chain with 6 edges, 12 segments. So the size of each edge is about 5 nanometers. So what we had to do is just to take this amino acid sequence, reversely transcribe it into the DNA, introduce this DNA into bacteria, and allow them to produce for us the protein, and then we characterize the protein. And we were quite thrilled that in fact, we did observe the desired polyhedra. As here you can see, I mean, they're very small object, 5 nanometers edge, but you can clearly see tetrahedral arrangement of vertices and also the shape. And then we wanted to go see how further can we get. Is just a lucky strike, or whether we can make more complex polyhedra by making the modeling platform where we can select in each of the selected shapes, and then we just design the topology, select the building blocks from our library of building blocks, make a model, and then go to the wet lab and isolate the protein. In fact, we went beyond tetrahedron, so here is the rectangular pyramid trigonal prism that's composed of 700 residues. It's all completely de novo designed residues, as you can see here, by small angle X-ray scattering. The shape fits pretty nice with the model, and also electron microscopy imaging shows that that's indeed what we designed. Further, we wanted to see if there are some potential applications. We are also sort of biased towards health, so ideally we wanted to use this for design of vaccines or something like that, and we showed that indeed those proteins are able to fold by themselves also inside human cells, but even in the living animals. So what's the main message to this one is that we designed a new fold, but it's not just a new fold that's not found in nature, but a new type of protein fold based on different principles. So in the natural proteins, the fold is defined by hydrophobic core that anchors the elements of the secondary structure of the protein. In this case, it's the topology, the order of segments that defines the structure. So ERC will certainly help to go in this direction. So what we want to do is to design more complex structures. In fact, the goal is really to design molecular machines by regulating the assembly and disassembly of those cages. And I think there are many potential applications: vaccines, drug delivery, enzyme encapsulation, sensors. And here, just at the end, thanks to my great team that helped in achieving this goal. Thank you.
Thank you again. Again, something that starts from very fundamental questions and can have important implications for health and other areas of human life. So we turn now to Silvia. Silvia is actually an indigenous person in this city, if I may say so. But she came back to Trieste after doing a sort of a tour of the world, from Scotland, UK to Australia, and back to Trieste. As mentioned earlier, Silvia is not an ERC grantee, but is an excellent researcher. She was highlighted in Nature. If you read Nature, you know what I mean. And she will present us her own work, after which we will start with some questions.
Thank you. So it's really nice to be here with you today. First of all, I think the main reason why I'm here is that I received recently this scientific recognition. So I just wanted to reflect a little bit on what I think this means, because maybe it can be a useful message for some of you that want to do science. So this is the link on the article. And basically, Nature Index wanted to recognize emerging scientists that have published for less than 20 years. So I think this is a message that we need renewal also in science and new ideas to solve problems that we haven't been able to solve as of yet. And what I didn't highlight here in the words, but it's also scientists that work on very relevant and urgent and global problems. So if you look also at the other profiles of these emerging scientists, I really encourage you to do so. You will see energy, health, environment, climate, and so forth. And where we look also at sustainability for what we can do as chemists. And then it's scientists that have initiative, curiosity, flexibility, and also a large network of co-authors. So really that have engaged in dialogue with different people with a different background and point of view. I think this is very important. So I'll just briefly mention my scientific journey, because I think a common trait of what we do that has been recognized is really the effort to cross borders between disciplines, but also borders between countries and cultures and nations. And I invite you, all of you, to do that, because you can learn a lot. So I started in Trieste. Trieste, I think well, you know, it's a beautiful city, so you'll see more of it during these days. And that's where I did my first my degree. And then I moved on with a PhD in UK between two universities. And then I did research in Finland. Finland is also very interesting. The culture is very different from Italian. They do a lot of saunas, and this is me doing an avanto. So you basically from 90 degrees, you go into the water, but outside in the air it's minus 20. So that's also an enriching experience. And then I moved to Australia, and you can see also here these are images of places nearby Melbourne and Melbourne itself. It's very diverse, and going down under really pushed me to think a little bit beyond the stereotypes and the limits of my imagination. I think many people, when they think of Australia, they think of tropical animals like these beautiful rainbow parrots that are indeed all over the city. But actually, just outside of Melbourne, you can also find penguins. So you really are forced to think a little bit beyond. And then, with all the luggage, cultural and scientific luggage and skills that I've learned all around, I returned to Trieste. And here we do a very multidisciplinary research. We are in the chemistry department, but we work at the interface between chemistry, biology, material science. We also use nanotechnologies. These are some of the keywords of the diverse research that we do. And I think probably we should switch off the audio, thank you. So during these days, maybe you've heard that we work on hydrogels. So this is really a video showing this hydrogel that is being made. What's so special about these materials is that basically they're made from very simple molecules, really tiny fragments of proteins. And then we spent a lot of effort in trying to understand how to convince these molecules to come together in ordered nanostructures. And when this system, and you can see here a video of molecular modeling showing that. And then when the system gets amplified over different scales, you actually get a macroscopic material, which has this nanostructure. The interesting thing is that these materials that you can see by eye, they're held together by very weak forces. So it means that you don't need a lot of energy to basically go back to the initial component. So you can do this sort of game where you form a material and then you assemble it and then you disassemble it. And so if the material with these nanostructures has a certain function, so it serves a purpose, this allows you to basically switch it on, do a function that you need, and then when the material has finished this function, you can switch it off and disassemble it into the initial components. A little bit like a lamp, you know, a light bulb. When you need it, you switch it on, and then when you're done, you switch it off. So I think also the concept is about a little bit good use of resources. And we do it in water. So what's the purpose of all this? Well, first of all, let's say that working with control over nanoscale dimensions is really important because it allows you to do things that you cannot do at a macroscopic level. This is just an example from Nature. Maybe you've heard of structural color. If not, I invite you to go and read more about it. So there are insects such as butterflies and beetles that have beautiful colors, but they're not the result of dyes as those that, for example, we use on clothing. They are the result of control over the morphology at the nano and micro scale. And then, of course, you get the effect at the macroscopic scale. And then we can produce dynamic smart materials that are water-based. They don't pollute because then they are disassembled at the end of the use. For what applications? We can envisage several applications. We're just at the beginning, but potentially they can be used for biomaterials, wood healing, drug delivery, therapy, but also smart coatings, even reactors. If these structures, they can catalyze a chemical transformation, you can use it to produce a chemical, to do a chemical reaction. So with this, I would like to conclude. And this is our group. As you can see, we're based in beautiful Trieste. These are the funding that allows us to do research. These are some of the faces of our collaborators. You can see lots of young people with new ideas. That's our website. And here are some of the names and where they're based. You can see they're very international. And so I would like to conclude with a message that I think that to innovate in science, it's really important to cross borders between nations, between cultures, and between disciplines. And I think we've had excellent examples also from the previous speakers that spoke today. So thank you.
Thank you very much, Silvia. This is actually, your conclusion is the question that I will ask each and all of you. But before doing that, I would like to ask a question, again, raising hands from everyone. Who of you was already at the session on the European Research Council yesterday afternoon? Okay, yes. So I will be very short. I was supposed to do a saluto at the beginning about ERC. I didn't because I wanted to give first the ground to our scientists. They are the better ambassador, the best ambassador of the European Research Council, and some of you anyhow heard of it yesterday. So it will be very simple and short. The European Research Council is a program to give space to innovative idea in the simplest possible way with a competitive and fair evaluation to get especially young scientists, this is where the largest part of the budget is devoted, to pursue their dreams, what they really want to pursue. The only evaluation criteria is excellence, which doesn't mean that we do not care about possible socioeconomic implications, but we believe that without great science, you cannot have great socioeconomic implications, let alone, for example, policy advice. So this is very much in short. If you want more information on ERC, there was a session yesterday, there will be documents and so on. There are calls for proposal. Any scientist after 2 years from PhD can apply from any part of the world, but with the idea to work in a European institution. Now with this, a first question from my side to all of you. Silvia was actually ending up with one commonality between all of you. Well, one of them is that everyone, which is very nice, ended up with a nice collective picture, because even if ERC grants and other grants are given to individual scientists to pursue their work, scientist is never a totally lonely enterprise. You always work with other people. In ERC, the average is to have around 7 team members for any principal investigator, which means in some groups many more, in other groups lower. But this is surely a commonality. Somebody gets the idea, but doesn't just develop the idea by himself or herself. And the other is that everybody has been crossing some kinds of boundaries. So the question is, what do you think has been the most difficult boundary to trespass in your work? Whether it was from disciplinary point of view, cultural point of view, geographical point of view, or others? And what was the biggest benefit you think you got in trespassing each and any of those boundaries? So I'll give the floor to anyone who would like to start first.
Okay, so I can, I can start. I think that the biggest boundary, the biggest challenge in crossing the boundary, at least in my experience, it was that I work with very different people, is the language. So I work with very different people, and first of all, we really speak different languages. And then we also aim for different things. But I do really like do this kind of things that I speak to the lay public. So that helped me a lot, because if you just downscale your language, everybody will understand, and also scientists will understand. So that's the main issue for me. And then the things when you don't speak clearly go a little slow at the beginning. But the best gain that you can get is that then they go way faster than you would ever imagine. You go to places where you even didn't think you could go. And also, and this is really important for me, you do have a lot of fun, because people from different disciplines think differently, and it's really fun.
Well, from my side, I think I completely agree with you, and I also had a lot of experience with working with people from psychology, cognitive neuroscience, and philosophers, so whole spectrum. And the curiosity and the fascination about certain questions that we have in our discipline, find out, we sort of need to. Yes, that's definitely a challenge. Another challenge I think that is there is actually dissemination of results in interdisciplinary fields, because there's it's very often difficult to find the right journals for interdisciplinary research, and we end up either publishing, in my case, it's either psychology or robotics, but then the work is interdisciplinary, so like the core psychology or core robotics sometimes are difficult to convince that these interdisciplinary questions are very, very important for bridging the... so that's a challenge, but again, as Laura said, it's extremely enriching to actually try to communicate to other disciplines why we find certain questions important, both in daily work and also in writing up papers into those and submitting them to various journals. So yeah, it's a lot of fun, a lot of challenge, and at the end of the day, it's a very enriching experience to work with people from various disciplines. Thank you.
So for me, the most difficult experience, I think, were the editors, really. I mean, it's you know, in life sciences, it's the fact that you have to publish in prestigious journals if you want to have an impact, even if you want to get an ERC. And we had a very hard time for the first paper that we published in Nature Chemical Biology. That was probably the 5th paper. It was rejected out of review with many journals. And then after it was published, they published the comments on this, complementing the paper. So I think that's the problem, that probably here in Trieste it's not so, I mean, the top university like Cambridge and MIT, so I think that's something that we have to work harder and be more successful than perhaps others. But I certainly want to end on the positive note. I mean, what was really the most rewarding in my experience is really this combination of mathematics and biological sciences. I also enjoyed mathematics while I was in high school, and I even now engaged with mathematicians actually even propose some theorems that were coming out of this one, and of course they also were excited. I mean, the mathematician contacted me when I presented this on national TV. He said that he made that type of theory for his master's degree, and he never thought it will be useful for anything, which also again shows that mathematics or even very basic things can be sometimes useful for something else. Yeah.
So I completely agree with the points that have been raised. The problem of the editorial wall, which I think is also connected to the problem of language. And the barriers, so connected also to the dissemination issue and to the barriers that exist when we try to communicate a message, especially with the other scientists from a different discipline or other people that do not have our point of view. So I found very useful the process of trying to identify what the barrier was and how to remove it. So for example, as a chemist, we use chemical structures to speak with other chemists. And with the time, I've learned that that's a barrier if other people do not have a chemical background. And the same goes for mathematicians, that of course they are used to use their equations, but when they interface with other scientists, often that's a problem. So I think identifying barriers and trying to remove them, that's a very hard job to do, but it's very rewarding.
Anyone has a question for any one of the speakers? Or do I have to keep going? Oh, there is a question over there. Is there a microphone? Or? Introduce yourself.
What?
Introduce yourself.
Okay. I'm a student of biological sciences, and I'm a bachelor degree student, so I know nothing. And I wanted to ask a question to the neuroscientist. I noticed that your robots looks like kids, and I was wondering if there are any kind of difference in attitude if you are dealing with a childlike robot or an adult-like robot.
Okay. Thank you very much for this question. Yes, I believe that the way the robot looks, the appearance plays a major role. And I can only, we haven't done systematic studies on comparing different types of shapes of whether it would be an adult looking like robot or a kid looking like robot, but I can tell you anecdotal evidence that we see in our lab is that very often people come to the experiments and they say, oh, it's so cute, oh, and then sometimes we even have effects such as the robot looks down instead of looking in the eyes, and people say, oh, it's so shy, the little robot, and then they start, very often they have this urge to pet the robot like they would pet the kid. So you just see, this is all anecdotal evidence, but you do see definitely certain attitudes, and I believe that if it was an adult-like looking robot, they would be different.
Thank you, Agnieszka. Anyone else who would like to ask a question? I see a gentleman over there.
Thank you. Thank you for the wonderful speeches. And I had a question for Professor Della about molecular modeling. You showed your proteins made basically by coiled coils, so maybe a bit rigid structures. Is it possible to build proteins not only made by coiled coil, but even with larger loops and more elastic structures, since it's quite, between brackets, easy to build them with software, but maybe through molecular modeling and through molecular dynamics, you could find that the structure is not as stable as you wished, and then in a physiological environment where the protein, I suppose, should work, then the structures get lost, and so you have no more the structure where you have planned. So is it possible to create even more elastic, more dynamic structures with the Nuove design?
Yeah, thank you. I mean, those are the questions that are in the directions that we are planning now. In fact, we have already made designs where we introduce the whole folded proteins into the loops of vertices of polyhedra, so they can address different types. But I think coiled coils will likely be the method of choice, because if you look at the beta strands, they don't have the time orthogonality, which means that you are not able to pair them so well. Besides, also in terms of the structure, coiled coils, it's long length-width aspect ratio, while with beta strands you usually have several strands together. So, I mean, dynamics is certainly something that we're exploiting. The molecular machines is one of the goals of the project. So to look at the dynamics also by changing the length of the loops, we have done up to now probably 30 different designs. And I think there are many, I would say sky's the limit. This time I may also add sort of an advertisement. We're still looking for the team. We have one Italians, but if there's a structural biologist for a postdoc or PhD position, you're certainly welcome to apply.
Perfect. So we are also doing a little job market advertisement, which is perfect. Okay, anyone else who would have a question? Here. Another gentleman.
Thank you. I would like to know from each of you if whether you're not going to decide who is going to benefit from your researches. If you are going to decide if this or this other community is going to benefit from the discoveries they mentioned you made, or someone else is going to decide for you, since you are researchers which get funds from someone else. Is this someone else deciding who is going to benefit from your discoveries or not?
Can I just have, I'm not the researcher in the room, well, as a background, of course, but the grants that are given by ERC, for those of you who were not in the room yesterday, are decided bottom-up, meaning we don't tell the researchers what they should do research about. We just say, any field of research you can cover. The choice is completely yours, and the method and the teams and everything, you just have to compete with the others to show you are really pushing the frontiers of research. And there is no requirement to demonstrate that the impact of research will be good for one or the other group in society. We know, it's part of science policy, that good research can, in the medium to long term, lead to sometimes serendipitous good impacts also outside science, but first you need the quality of science to have those impacts. So this is the philosophy of the European Research Council. And the issue of independence of researchers in choosing how they go about the research is actually fundamental for us. So I think it's a great question, but I think the researcher should then now explain both with regard to ERC grants and non-ERC grants. So maybe we start with Silvia this time.
So in my grants, I do not have any, let's say, I do not have projects with direct industrial involvement, because, for example, industry may be happy to invest in research, also with the idea that it could benefit a certain industrial process, so something that also goes directly to the public that needs a certain tool or a certain process. So the research we do is a little bit more fundamental, and basically we have funding from the Ministry of Education and Research and from the EU, mainly through cost actions that promote a scientists' exchange. And so, in terms of benefits, I think is the public, but it's something more like the longer term. So, personally, I can say I'm very open if people come to me with, they think that my technology can be useful for a problem that they're tackling. It could be an industry, it could be an investor, I don't know, it could be a billionaire that wants to invest in research. And then we are happy to look at a more specific problem with the help of people that work on that problem and have more specific knowledge. And then the benefit would be, for the people that problem is relevant to. So it could be people that have a certain disease, people that are involved in an industrial process, it depends.
But starting from the independence of the researchers who will accept or otherwise what they do. Any comment from any of you?
Perhaps just to add that it's open science. So the ERC is done under the open access, so the results are available to anybody. And I think most, as mentioned before, ERC is not really intended to do specific applied research, but sort of the blue sky. And honestly, I really don't know what I will do in 5 years. And I think everybody who is doing in some exciting field doesn't really know where it will lead. We have some certain directions, but I think each researcher is just very happy if some of its research is applied for something useful.
So I think I can only add the fact that when a funding agency wants to fund some project, they open a call and you are the one that proposed the project. So you are the one that is in charge of what you're doing. And usually it works like that. Then who decides who is going to benefit are the people that are going to benefit. So they will sort of reach at you and tell you, oh, can we do something together, as Silvia was saying. I only had one experience of a funding agency that kept rejecting my application and tried to, mold a little bit of what I was doing, but you're always in time to say, okay, I'm not going to apply. So nobody will apply.
Okay, so I will...
the working.
Okay. Who would like to achieve and what you actually achieve?
If I understand correctly, I just want this fantastic job. I mean, it's very difficult. You have to be aware that doing science is not an easy job. It's like 8 hours a day, but great science, really dedicated. You work with, I think, for science, I think
the moment you run the experiment, you have questions, and that is the gap between sort of expectations and reality. Science forward. So.
Anyone would?
The other thing that I would like to add is that I think so. There may be a certain gap between what you expect to do or to achieve with an experiment and what you actually get. I think first of all, it's important not to what you expect, because that can. But also I think that it opens new questions, experiments that you want to get.
If you want to push the frontier, your expectation must include the fact that you might fail and that you need to have a plan B. Either you fail and you get to new questions, whether it is about theory or it is about experimental science. So if you do expect that you might fail and that you learn from it, and you can eventually change or adjust your area, well, then I guess that expectations are not the biased one that get you completely frustrated and then you stop what you're doing. It's just, keep you open to, the path of discovery, which is hard. Yesterday, somebody asked in the previous session, what are the recommendations for young researchers? One was work hard. The second was trust yourself and keep your passion and curiosity. So I think with this we close. Antonio is back with us. Thank you.
Thank you for an inspiring talk. Unfortunately, we have to leave the room because there is quite a crowd outside waiting for the next event. So thank you very much to our guests. It was really an inspiring talk, and I know that there are some questions up there, so you might be available out of the theater hall to answer everyone. Il prossimo appuntamento è fra 15 minuti, Scienza, Etica, Società, con la professoressa Elena Cattaneo. Grazie mille a tutti e grazie ai nostri ospiti.